So AT, or ataxia-telangiectasia, is a genetic disease. It's caused by inherited mutations in a gene called ATM. Now we know, and have known for many years, that patients with mutations in ATM get the disease ataxia-telangiectasia. This is a multifaceted disease, but most notably these patients, often in childhood, can end up with cancer, or even more importantly, neurodegenerative features. And what this neurodegeneration is that certain cells in the brain are prone to die. And most of those nerve cells that die in AT patients are those nerve cells that are involved in controlling coordination. So one of the real problems with this disease is general coordination difficulties that get worse and worse with age. So that is the basis for AT. But another thing we've known for many years is that different patients with AT have differing degrees of severity. Some patients have milder disease than others, which suggests that it's not just the ATM mutation itself, but maybe some other features about the individual which makes them more or less likely to develop very severe disease features. It turns out there is one patient here in the UK, a lady who has a very severe ATM mutation, 2 mutations in the ATM gene, but has very mild presentation. So this patient looks like she should get full-blown AT, but has very mild features. And the overall concept and goal behind this project is to see whether or not there's something special about this lady's DNA, something special about her genome, and therefore special about her cells and herself, which means that she's got some resilience to AT. The project involves genome sequencing, but to explain genome sequencing I need to first explain to you what the genome is. The genome is a term given to cover the entirety of the human genetic information in the DNA. So DNA contains sequences, it's various letters of a sequence, a very complicated sequence in human cells, which basically carries the code for what we are. It tells our cells how to work. So the genome sequence of a human is this information that makes our cells behave in a normal way. Now it turns out that corruptions to that sequence, changes to the sequence, called mutations, can cause cells to behave in the wrong way. And mutations, for example, can underlie genetic diseases such as ataxia-telangiectasia, AT. So what we aim to do with this project is to use genome sequencing to understand the genetics of AT, and in particular try and see whether or not there are certain other genes that might influence how severe the AT disease is. So genome sequencing is fairly easy to understand at the overall level, but the challenge is that the human genome is huge. There are around 3 billion letters in our genome sequence. And just to put that into context, if you were to put the human genome sequence as letters in a standard book, that book would have over 1 million pages. Well, first of all, I have to point out that this is basic research. We don't know what the outcomes are going to be. But the goal of this research is to try and identify genetic factors that may influence how severe the AT disease is. So at the least, if that works, we might be able to start to understand why some patients get the disease more severely than others. But even more exciting than that is the possibility that this research might identify a way to alleviate disease, alleviate the ataxia-telangiectasia in patients that get the full-blown disease. For example, if our studies at the genetic or the cellular level identify something that we could one day develop a drug against, then that might provide an opportunity for developing drugs that slow down or ameliorate AT as a disease. Research into AT is, in my view, extremely important. First and foremost, this is a devastating disease which strikes people down often in childhood, which has devastating consequences for those people and their families. This type of research offers the potential to explain the disease better and maybe identify ways of slowing down or alleviating the disease. Secondly, it's clear that AT provides a paradigm, a model for other related conditions. There are other inherited diseases with similar features, so that what we learn for AT might be applicable to these other conditions, and of course that might lead to health care improvements for those other diseases. And thirdly, it's now becoming clear that diseases like AT, studying them, provides models that can be used in much broader contexts. And it's my view that if we understand diseases like AT and understand how neurodegeneration and cancer can occur in these patients, we're also going to have a much better understanding how neurodegeneration and cancer and other features can arise in more normal patients in the broader population. So by studying, if you like, rare diseases like AT, the impact is not necessarily restricted just to those diseases. There's the potential for this to open up healthcare improvements to much wider populations. Action for AT is playing a very important role in furthering our understanding of ataxia telangiectasia, AT, and related diseases. The charity is very active and, of course, is active in supporting cutting-edge science such as that which is being carried out in my lab with our collaborators. So Action for AT is making a real difference to research labs like mine, helping us to carry out this, what we think is very important research, which may lead to improvements in our understanding and hopefully treatment of AT and related conditions.